developing.
Tyler just asked me to record it, so I'm going to record it for the team.
Thank you. Thank you. Yeah.
Navakant, should I go? I would like to add. So this is Krishna here. So we are currently stuck. So there are two approaches which we are discussing. One is the paper microfluidics, as Navakant said, and the other approach is the using optical based sensors. So we are currently figuring it out which is the right way to go. Because in case of you, if you go for a optical based sensors, we were thinking. So for example, if we want to detect nitrogen, phosphorus, and potassium in the soil, there was a paper where they said that if you have a different wavelengths of light, so based on the absorbance, you can detect the concentrations of the nitrogen, potassium, and phosphorus in the solution. So we were thinking maybe we can implement it using a smartphone. You will have a flashlight at the back of a smartphone, and then if you have a small filter, and you can choose the wavelength, or you can use some wavelength and detect the content of the soil. So that was one way to go. And the other aspect is the microfluidics, where you will have a small piece of paper, and we will have a pattern, a flower like pattern, wherein if you just add a drop of soil sample solution, it will go into different directions, and wherein you can get different colorimetric values of which are related to the concentrations of the soil. So the place which we are stuck is which way to go. Yeah, go ahead. Yeah.
My understanding is that you need a reaction, or at least nitrogen in water and potassium in water and things like this. Often it's a calorimetric reaction. You have to have it in a fluid, and then you look at changes of absorption, for example, as you're adding a reagent. Is this the direction you're going, or are you looking at, taking a picture of a soil and from this inferring the composition?
No. So what do you mean by the taking the picture? Means just take the picture and then look at the concentration? Is that what you meant?
I try and infer concentration. I'm not clear about what approach you're thinking of getting. Are you gonna take soil, put it in a fluid, extract something out of it, and then do reagent? Put in reagents to see change in color, or?
Yeah. That's the approach we are looking. So you just take a soil and then put it in water, just shake it, and whatever solution you are getting, and then you use it for the further reactions. In case of microfluidics, and maybe for the same for the optical based sensors as well. So we don't have expertise based on the optical side. I think so. I can see somebody is sharing this thing. So we don't have expertise in optical based sensors. We have expertise in microfluidics, and Brenda, I think so, she is there. She's on field. She has experience with dealing with the soil samples. She directly works with farmers in Brazil, I guess. And then we have one more team member who is involved on the field. So we have people who have some knowledge on soil, but we are looking for also chemistry. So we are looking for optical and maybe electronics plus chemistry, more on reaction chemistry as well, through which if we can find some chemistry. So that is one more issue, that there is we have to look for a specific chemical reactions through which we can determine what concentrations we need.
We also need a designer.
Yes, that's true.
So from other mentors in the group, anybody wants, and others in general, all of you can mentor each other as well. Any suggestion of things you may want to propose to this group, given where they are?
Yes, so one approach is possible that people use in the farmers in India, especially the institutes that have the farmers in India, is basically they have this chemistry based kits with the reagents. You basically go there, pick up soil, and extract using water, and then basically the concentration is against a kind of a gradient, and then quickly plot it, so you get a good, pretty good value across different macro and micronutrients, including NPK, other metals. The other, in fact, Government of India now does this kind of soil health cards that are
Yeah.
funded.
Yes.
So the other approach, and it's kind of covered under your optical way of using it. There are many companies that are now trying to use the satellite imagery to do this kind of detection at scale to bring down the cost. So maybe some of papers that would allow you to play with, understand some of the complexities, or whatever the spectrum and other computational complexities from those papers. Probably those two different ways. And probably chemistry-based ways where you have this actual reaction, color change. It's pretty well documented, and probably that might help you cut down the cost pretty quickly.
Yeah. Thanks.
Okay. Microplastic detection. Sorry, microplastic detection group. Just so we get to talk to everybody because time is running.
Okay, can you hear me?
We can.
Okay, this is Srinikita here from Microplastics Group. I will briefly tell you about how our journey been into the research. So first, we have started doing a literature review. So we boiled it down to three ways to approach this particular microplastic detection problem. One way was optical. The another way was spectroscopy, and the third way is capacitive sensing. So actually there was a lot of literature that is on Raman spectroscopy. So we have been doing literature review on that, and we started doing couple of meetings regarding that. But in our last mentor session with Manu, we have been introduced to impedance based detection devices to build upon. So generally in the literature, it is being mostly used to study cells and the molecular properties of cells. So now we want to use a similar kind of device to sense plastics and understand their dielectric properties. So, and while I was listening to John Vale, sir, like in our team, we do not have anyone who are expertise in electronics and electrical devices. So we would like your help. So,
I'd be happy to help however I can.
yeah, any comments?
I mean, just sort of off the cuff, it certainly seems like a good approach to try to measure things using dielectric properties, because plastics are very far different from water in that regard. So, I mean, dielectric constants of water is something like 70, I think, and plastics are going to be far, far less. So that should be a good way to approach in general. But I mean, of course, there's lots of details as far as the specifics of how mechanically that would be done.
Okay.
So did you think about how you're going to isolate the plastics from the environment? How are you going to sample for plastics?
Like actually we have chosen inland waters like rivers and lakes. That is where we are targeting. So like our target audience is anyone who has access to technology. So we wanted to make a frugal technology accessible to anyone who is interested in looking after inland waters like rivers and lakes. So
but do you expect them to filter water and concentrate the plastic, or you want them to measure it in the environment without concentrating the particles and just looking at particles passing in front of you and then saying, this is plastic, this is algae, this is sand, this is, what's the direction that you're going in terms of?
That we haven't decided yet. So we are still figuring out things.
because plastic are relatively dilute in the environment. I mean, you don't
Yes.
see big mats unless you're in certain places and it's a conversion zone. So you'll have to decide whether you want to concentrate them before and do a bulk measurement versus you want to do individual pieces properties.
Yeah. That we are yet to decide in the team.
Any other comments from anybody?
I guess probably your suggestion about figuring out microplastics, either kind of concentrating it otherwise, or kind of just some way to get some microplastics and some sort of filtration going on, maybe. Then kind of trying out the very controlled experiments with your dielectric constants. You have water, and now you have a concentrated microplastics. You can make your own solutions and then kind of test it out. But yeah, given you have to first show that given water body has microplastics to begin with.
Yeah.
And I would add a little more to that. There's plastics fluoresce. It's one of the way by which they're separated in recycling facilities. So you can detect specific excitation emission period that go with specific plastic. That's once you concentrate it. And also there are methods that size a particle using resistance and like Coulter counters. And those will be very sensitive to the type of particles you have and its dielectric constant as it passes through a conductive kind of cylinder, which might be a way. This is if you want to do individual particle counting as opposed to spread them. And one method that people have done with concentrated particle is to spread them like in a scanner. And then they scan all the particle they collected. And then if you look at fluorescence versus other properties of whatever you spread, you might be able to separate plastics.
Okay.
Should we move?
Before we move on, I was just going to note that I did throw a link in about a plastics kind of a trawl processing from Public Lab a few years ago that might be just worth looking at that whole thread that they've got there.
Okay.
Thank you, Pat.
Thank you.
Should we move to the locust swarming group?
Hi, thanks. Yeah, and I'll keep it quick because so other teams have a chance to go and we're pretty early on. But obviously this locust swarming issue is a continental scale issue. And for people who are not aware, you have these locusts that contain hundreds of millions of insects swarming across very large swaths of land. And so that's a problem, obviously, that requires maybe government-sized entities to really solve. But what we're interested as a team is focusing on if you have an individual farm or say a small tribe or a small group managing a farm or livestock, what can they do if this problem is right in their backyard and affecting them locally? So we're very early on in terms of right now what the stage our team is at is trying to interview people in various countries who are impacted by this and understand the locusts and their swarming behaviors better. One thing, we've found a little bit of scientific literature in controlled settings looking at locust behavior in terms of whether they are attracted or repelled by different sensory stimuli. And that's something that we're interested in studying more. Obviously, so right now, for more context, what's mainly used are pesticides, but these obviously have a big negative impact on the environment as well. For example, if you spray down your entire field with pesticides, then maybe the locusts will be gone, but you have that ecological damage, obviously, and sometimes livestock get sick from eating the pesticide-ridden plants. And locusts, again, for reference, they eat pretty much any vegetation. So one idea that we have generally is, okay, how can we trap these locusts or most of the efforts right now are into, I guess, repelling locusts. So it's pesticides, and people bang pots and pans, for instance, to try to just get them away and get them off the crops. And they're constantly walking through their fields trying to remove them. And we're wondering if you flip that, if you try to attract the locusts to maybe one part of your field over others and sacrifice even half the field for the other half, which would be an improvement upon the status quo. So we're interested in, well, there's two steps to this. One would be what sensory stimuli can attract locusts, and how would we find that data, which could look right now like, we've looked a little bit into literature, but also I think it could be interesting to try to crowdsource this kind of like information. If people have any advice or thoughts about that kind of idea, like trying to get actual data from people on the ground dealing with these swarms, which I think would be very useful, especially compared to like controlled studies in a controlled lab environment where you're not sure if the locusts are behaving the same as they would in a gregarious swarm of many million. So if people have any ideas on that, that would be very helpful in terms of gathering this data and how we could facilitate that. Let me think. And then the second part of this problem is if you can attract them to a specific place, then you need to deal with them in some way, whether it's with concentrated insecticides or something else. And hopefully though, you would do it in a way such that you can use these locusts potentially even for a food source or for chitin, something else like that when you collect them, some product to mitigate the disaster that they're wreaking on the environment. So if people have ideas as to how to contain locusts, once you have them in a certain position, like early on, we thought about biomaterials, like something like a sticky gel or something like that, that would be edible at best, or just less potent than these pesticides. Any kind of improvement would be interesting. So I know that it's this problem is very large scale. So the type of expertise we could need could be from all over the place. So if anyone has any ideas, just feel free to drop it in the chat or send us a message, because we're open to anything right now. So thank you.
Any comments from anybody?
I guess one source of data that uses this kind of light and sound show is probably USDA, not specifically they might have for locusts, but there is some interesting research that has been published that fields that are nearer to highways and where there is kind of soundproofing not available, those fields, because of the sound and light, there are overall less bugs in that area. But maybe it kind of tapers off as the field kind of away from the highways. Maybe kind of also tap into the insect eye structure that probably kind of there are certain wavelengths of light that could be really annoy the insects and just like complementing the sound method to kind of annoy the insects away. Certainly, I think that kind of concentrating them in the area and kind of using them for source of chitin and whatnot, I think is a great thought. Going back to the kind of light and sound show, there's quite a bit of literature, especially published by in Japan, who have used or at least have done this kind of controlled experiments. Not again, I don't recall locust as particularly one of the, but there are other bugs that really could cause havoc on the agriculture side, and they are definitely studied, and they have lots of ideas and suggestions in that. So that's probably in the literature to look at. But certainly, whatever I have collected on the light and sound show with respect to insects, I can definitely forward that to you.
Thank you. That would be very helpful if you can forward those links.
Thank you. Should we move on to the toxic algae group?
Yeah, sure. I'll be speaking on behalf. So currently where we're at with it is we're still trying to kind of identify the problem specifically that we want to target. So our understanding is that in freshwater environments, cyanobacteria are very prevalent as opposed to kind of marine environments where dinoflagellates are more common. So trying to understand which kind of species we're looking to kind of target and detect early on. I think having a broad scale solution for all of these kind of blooms is going to be very difficult. So trying just to kind of focus on one thing. So we've been pursuing two lines of thought, and at this point trying to decide, do we want to wrap them up in one bundle, or do we want to kind of make one that can kind of accomplish both ends of the spectrum we're looking to do. So one is like a cheap fluorimeter, which I know Emmanuel, you sent this paper, and that looks really promising, just to figure out which excitation emission pairs are going to show up in these blooms, and then detect them using that type of technology. Another thing we thought about is repurposing some kind of paper-based chromatography. So something even as simple as TLC, linear chromatography, that can just detect quickly and rapidly whether there are maybe some molecules that shouldn't be in the water that we might be wanting to look at, such as the toxins that are in these harmful algae blooms. But at the same time, it might be more simple and more effective to really focus in on fluorimeter versus trying to mix and match both of them together to get something. So that's kind of where we're at with it right now. We are still very much in the design phases of it. And can we, how do we want to have this? We want something that's modular that we can deploy to people that they can use, or maybe some kind of floating device that can kind of be statically out there to detect them as they come in. So any thoughts on that in that realm would be greatly appreciated. And I think as far as expertise, like I said, design would be huge, as well as just general kind of marine electronics. I think I personally don't. I think Ben has a lot of experience in that realm, but so anything like that would be. We'd love to hear thoughts.
Have you seen the Plankton School project from our lab?
Yes.
Yeah, so I'm thinking something similar to that, where you have kind of a chamber and that you can flow. You have a continuous flow, so it's a continuous monitoring. So to see what you're coming against, or something like that might be of interest.
Great, thank you. Write that down.
Other optical techniques, there's spectroscopy, simply looking at their absorption signature. We know they have very specific phycocyanin and phycoerythrin, which have specific absorption bands. And the second one is remote sensing. And there's a huge amount of work that's being done now, but it's not deployed yet globally the way it should be. So satellites like Sentinel-2, A and B, they revisit every 5 days between them. And Landsat 8, they're very high resolution, relatively speaking, so tens of meters. And they're already algorithm to detect presence of cyanos in the water with those. The problem is that they're not yet deployed globally, and not on every, you'd love to have them deployed in every water reservoir, every lake, every place where they might occur, particularly in the third world where they don't have the expertise necessarily. But the data is available, it's free, it's there. So it's maybe even just making that data much more accessible to people.
And that was Sentinel-2, you said?
Yes, Sentinel-2.
Great, yeah, thank you.
A and B, there's two, A and B, but they're the same. They just revisit.
Great, perfect, thank you.
Any other comments? So I think our last group, there's two more. There's the ant family and alternative packaging. So ant family, can we hear from you?
Yes, sir. So we are from ant family group, and what we aspire to do is to monitor the environment using different species of or the diversity of ant present in a particular area. So why it can be really helpful is that everybody can differentiate that whether this is an ant or any other insect. Right now, what we are doing is we're working on how to identify ants. And for that, firstly, we are studying the morphology. And secondly, we are using the foldscope to capture the different parts like antenna or the petiole, the thorax, abdomen, and then trying to identify it using the keys and online sources like iNaturalist and all. What we are aspiring to do is to make it into a citizen science program, but for that we have many hurdles, like how to capture a good picture of ant and then to classify it. So for now, what tools we are using is like lens, but again, one problem with lens is only experts can use it and directly look for the parts we want to study. And using all the collected data, if once we collect the data, we are planning, or maybe if we come up with an app to identify the ants. And but again, the main hurdle is to collect a good picture of ant, and simple mobile phone cameras cannot do that. For now, what we require on our group is an ant expert, the person who has worked with ants and know them quite well, so that firstly we can at least learn how to classify them. That's our big hurdle.
Any ant experts among us, or taxonomists, or somebody who knows ant taxonomists? Or where would one go and find I mean, I assume what you do is a literature search on ants, and you look at the researcher and try and find one of them who has the will and the time and the interest in joining force with you to help you with the taxonomy. I think you have the expertise here for doing AI once to help you, once you have a lot of images, classify them using machine learning. But as you're saying, yes.
I'm from the ant family as well. So in the 15-minute meeting that we had with Manu sir, he suggested that we assigned a mentor named Lucas Venezuela, who has an experience in machine learning and AI. But we have also, after the discussions in the subsequent meetings, that we also need an ant expert. Because one suggestion that was made with respect to AI was the use of Ciliot. I don't know if I'm pronouncing this right. The use of Ciliot for ant identification. I don't know what that means, but we would like to explore that. So these are two basic requirements. One is expertise in machine learning and AI for the app, and an ant expert.
Sounds like you're on track. We have two more groups. We have one is the alternative packaging, and we have the space debris. So can we hear from the alternative packaging?
Sure. I'm Rishi, and kind of speaking on behalf of the AltPack group. So what we are currently engaged in is building a database or list of biodegradable materials that we want to compare and contrast with plastic across, say, example, physicochemical properties, elasticity, molecular structure, and others. Because plastic is widely used, so it satisfies a whole lot of criteria that industry demands. So we would want to know what those standards are, and also, if possible, perform measurements that kind of generate some early data that can be compared against, and see if it meets the beats plastic or falls really short, or what would be an appropriate area based on these kind of measurements or properties. And also, we are hoping to engage with institutes or corporations under like an open innovation model that can use this early insights into the measurements and kind of engage more in a fruitful manner, and so that we have an end in sight. So the current requirement where we can use help is about what those standards are that plastic meets, and what are the kind of measurements, what are the units and dimensions, and how does one test that, and so that we can do a vis-a-vis comparison if possible for any of these biodegradable materials. And we have started compiling the list, and we have for example, like palm frond, onion peel, deciduous tree leaf, and see where it can be used. But we definitely need measurements, help with the measurements, as well as the standards that the plastic satisfies and makes it itself attractive to the industry.
Excellent. And this links to another group which I named but forgot to call on them, which is the reduced lab waste group. So a member of that group, do you wanna kind of tell us what you're doing and how potentially sounds like there's a synergy between you and the previous group?
Yes. Hi, everyone. I'm Kavya, and I'm speaking on behalf of the Reduce Lab Waste team. Can I share my screen just to show where we're at so that it's easier to follow? Thank you. So as of now, we have a few things that we have decided with respect to what solutions we want to have. One of the major things that obviously got talked about is biodegradability. But after like having a conversation with Manu, like last week, I think we realized that is one of our low priority, because this would require collecting material information and designing microtips, and everything would not be feasible in 2 weeks. The solution might not be feasible in 2 weeks. So another set of idea is 3D printing. So here, the current idea as of now, suggested by one of our teammates, is to reduce the plastic amount in the existing microtips by having two layers in one microtip. And this would either be a biodegradable plastic, or we are yet to figure out what it can possibly be. But this could be something that's reusable or like ejectable. Essentially, we would be reducing the amount of existing plastic in the microtip. We are yet to see, and we are yet to vote on if this is a feasible solution too. And the plus point would be we would be incorporating biodegradability. The negative point would be that it would we are still not answering what would happen. We're not still posing a solution to what actually happens to the existing plastic that's there in all the labs. Coming on, which brings us to our third idea, which is okay, we just have 7 minutes, so I'll be quick. There's something, so this idea is just to have a super rinser, and this would be by redesigning Vortex. In case anyone doesn't know what that is, this is what it looks like, and it helps you shake things up pretty quickly. So we were hoping to redesign Vortex into a small like washing machine to wash off these microtips that are existing in the lab, and even this might even work out for the Eppendorf tubes, for which the solution, the ideas would be to figure out protocols which we think will be easily available, and redesigning Vortex. That would be a major bottleneck. The next one is a solution called wax molding, which will be explained more in detail by Emily. But before that, we have data collection here, where we have to get info on what kind of plastic is mainly used in lab and what kind of waste is generated by lab. Is it tips, Petri dishes, Eppendorf, which has a bigger priority, and which will help us narrow down to what to focus on. The other one is what kind of experiments are done with these plastics and these materials, just to see if reusability is more feasible than redesigning the material using a biodegradable thing. So our Notion page is here, and these are our ideas with respect to this. Maybe Emily can talk about wax molding a little bit more in detail.
Let's hold on this, because we will not have time to get back. Just is there specific expertise you're looking in this group to get information on, or people that you want from this group that can help you with something? Is there something that
we actually would like more of a material science background with design understanding, and probably electronics, and probably electronics too, and machines in general. Because like if we are thinking about Vortex and redesigning that, we would like some mentor who can think about how
that will be excellent once you focus on one of the things that you're gonna focus on. As you said, the time is running out for everybody, so it would be really good if you decided on the one thing you're going after, and then call Manu, and he'll propagate it to the rest of the mentors and for the specific thing you want to do, if it's the washing machine, if it's the double tip, and etc. I think that will help a lot. Any other comments from the public before we move to space debris? So let's go to space debris. And there's a lot of, there's wonderful stuff on the chat. So all of you monitor the chat to things that are directly relevant to you. There's wonderful suggestion there. So we have several conversations in parallel. Yes, space debris.
Yeah, this is Phil. I'll be talking to space debris. So there's hundreds of thousands of small particles in low Earth orbit that are hazardous to expensive satellites and even manned space flight. So we're looking at trying to come up with lower cost ways to mitigate that kind of a problem. So the first step is trying to characterize what that environment looks like in terms of what's the density of the material at what altitudes and at what orbital inclinations. And once we have that characterization, we're looking at that as helping to us to assess potential solutions. For example, one thought is to use something like a big sheet of flypaper, kind of think of it. That could be, I don't know, 100 yards by 100 yards sheet that would sweep through an orbit and gather up all the garbage. And then at some point when it's saturated, you deorbit it. But that may not be effective. It kind of depends what the density of the particles are. Another approach that's gotten some literature is using focused lasers to target the debris, either burn them up or cause enough momentum change that they would slow down and fall into the atmosphere and burn up that way. But anyway, some of the places we're looking for help is to get some help with the analysis and simulation tools to try to model what these orbits would look like and how these potential solutions would interact with the debris to clean the stuff up. We've started to make a contact with one company, a commercial company that sells as a subscription service to satellite operators, kind of collision avoidance, because they've got some simulations and models. And so we try to reach out to them, haven't heard back from them yet, but we also be looking for other help that could help us model what the debris looks like, as well as simulate what our solutions impact would have.
Sounds like a very important problem. Anybody has anything to contribute?
I did a little project on space debris a while ago, and I wanted to show out a few things. If you check the NASA website, there's a tool called ORDEM, O-R-D-E-M, and you can request it. It's a bit of a hassle. You have to fill out a form, and it will take a while, but they will grant your request unless you're very suspicious. I have a weird IP address or something, and that tool gives you data on flux distribution at different orbits at different sizes, and it's super helpful to kind of tell you about what flux you can expect at different heights of different particles of different sizes, and they take into account both the track catalogs for larger debris, but also measurements of particles that are too small to detect, where they send something into orbit and counted impact. So they're pretty cool, and they have data that goes back into the past, and they also predict the future, because obviously Kessler syndrome wise stuff is gonna just get worse. That's a really great tool. It's a bit annoying to get used to, but that's a great tool.
ORDEM, right? ORDEM.
Yes.
Cool. That's great.
Right.
We have to go back to the general room. We just got a message from Tyler saying to wrap up. So thank you, everybody. I think this was very helpful. I hope it was. And Amanda and Phil, if you can touch base directly, then you can exchange. I'm sure Amanda is much more about the subject than.
Thanks, Amanda.
And so there's a link down below. It says Live Room. I think this is what we have to click on to go back to the full class. So see you all there.